This method illustrates how the combination of temporally and spatially discrete in vivo electroporation with transgenic lines of zebrafish can selectively target regions of the developing embryo for transgene expression. Begin with Gal4 transgenic fish that were derived by the enhancer trap method to restrict gene expression in cell types of interest, in this case the developing olfactory bulb. At a specific time in development, inject the GFP reporter plasmid into the anterior end of the forebrain in vivo and perform electroporation to facilitate DNA delivery. Results obtained by in vivo fluorescence imaging indicate precise localization of the transgene at a specific time and process of development.
The main advantage of this technique over existing methods, like injection of plasmids at the one-cell stage, is that expression is targeted to specific cell types within a specific region of the developing brain.
Use a transgenic line of zebrafish created through a transposon-mediated enhancer trap method to drive expression of both the transcription factor Gal4 and the reporter protein mCherry at specific regions of the developing brain. Set up mating of adult fish heterozygous for the transgenic insert with wild-type fish to yield approximately 50% transgenic offspring. Now treat transgenic embryos with 100 micromolar N-phenylthiourea beginning at 6 to 8 hours post-fertilization to block pigment formation and facilitate fluorescent imaging.